Tanja is a Worcester Polytech Associate Professor of Biology and Biotechnology and a 2011 Coleman Fellow. I try to give you something different than what's written up in your program. And she is a prime example of how academic research and entrepreneurship go hand in hand. And at the very first meeting that we had of the Rosalind Franklin Society, we found that most women who were in academia were not interested necessarily in collaborating in any way, being on the boards, or starting a biotechnology company or moving in that direction. However, it turned out that when they heard more about what board positions might be, they became more interested and were interested in seeking positions on corporate boards. But going into biotechnology, there are very few women who have started up a biotechnology company successfully, and Tanja is one of them. She is the founder and CEO of a company called CellThera Incorporated, and she's going to tell you something about it. She teaches, by the way, an undergraduate introduction to biotechnology, a graduate level course in cell cycle regulation, and of course, so you can see that there is an extraordinary mix of talent, ambition, and capabilities here, and hopefully she will inspire more of us to look into the academic and industrial opportunities, because as industry grows, it funds research, and it takes the research and hopefully produces products that are going to be beneficial for all. Okay, Tanja? Here you are.
So I think we were all given titles for our presentations, and we were all, some of us more, some of us less, surprised by Carla, that you're doing, I think. So I said, well, I have to put this talk together, and I didn't even know what my title was. So I'm looking Plasticity and Regeneration of Cells. Yeah, that is part of my life. A Coleman Fellow entrepreneur, that's another part of my life. And then it really got me thinking about if I had to summarize my career, or if I had to pull highlights out of my career, would this be really it, or would I add anything new? And so I decided to approach this talk much less from the scientific point because of the audience, but also because I think this is really a great opportunity to share some of the unique stories that we have heard here. Maybe add some humor to the path that some of us have walked, and maybe finish with something a little unique that doesn't have to do with science and technology. So I tentatively titled what I would like to tell you today, hopefully in 15 minutes, not more, about how I am redefining how I see my success as a professional and as a person. So this is where I started, is way on the top. This is what I would like to tell you about today. Hopefully this path is not going down. I hope it's still going up. So if I could, I would change the direction of it, so it would still continue to go up. But I'm a veterinarian. I got my veterinary medicine degree at the University of Ljubljana in Slovenia. Came to graduate school to University of Wisconsin-Madison, and did my PhD. And I think tried to summarize, and you can't see it, what pretty much a successful scientist looks in United States and what defines us as competitive, maybe researchers. I have had funding from DARPA, NIH, NIST, USDA, all competitive funding. I have published also in Nature and Science. No, I have not published in Cell. Hopefully that's coming sometime. I sit on editorial boards, quite a few of them. 3 of them are on journals from Marianne Liebert. She too, to me, just like Sherry said before, has been a great mentor and a friend over the years. And I sit on a review panel. So I think I have rounded my career kind of nicely with the package of accomplishments that I have. I'm also an educator, most of us are. I'm an associate professor, teach a number of courses at WPI. And the first part of my title, you saw entrepreneur. This was really more by an accident than by a desire to be an entrepreneur and a businesswoman. I wanted to follow research that I was passionate about and was not able to do so in the current environment, so I decided to start a company. I was in my pajamas having morning coffee, and I said, I will have a company, I will get a tax number, and then I can compete for my own funding for what I want to do. So this was really not this great foresight, how to become an entrepreneur, but really to enable me to pursue my own research. Luckily, I have been fairly successful in doing that. The company has been around now for 9 years, and we are developing strategies for autologous cell therapy. I will tell you a little bit about the biology of it. Coleman Fellow Entrepreneur, I was accepted into the program that's run by a foundation in Chicago, and the purpose of the Coleman Foundation is really to try to infuse as much as they can entrepreneurship and creativity education into undergraduate and graduate curriculum. So it's recruiting faculty that teach in a lot of engineering departments, science departments, humanities departments, and try to develop strategies. How do we convey that no matter what field we are in, there is a way to be creative, there's a way to be innovative, and most often, some of this has potential value for the larger community. So I'm trying to get really creative. How do I teach entrepreneurship in cell cycle? And maybe it's not obvious if you've never tried to think about it this way, but clearly we have heard today that developing new small molecules or small drugs for cancer, for example, that are based on cell cycle. We have had a great lecture on Taxol. It's probably one of the things that can come to mind when you start thinking, okay, what can we do to manipulate cell phenotype for a variety of reasons? And again, I don't want to spend too much time on this. Of course, I am a community volunteer, like we all are, school board member for 7 years. I have 2 daughters. I'm a proud mother of a future physician and a future oceanographer. I hope that still happens. She's still junior in high school, the second one. And with that, of course, all the PTA meetings and PTA organizations and teacher conferences and everything that goes with it. But I really, through all this, I have really this year decided that I need more. Not because I'm not busy enough, but I need something that will more define who I am, and something that maybe in 20 and 30 years I will nostalgically look back on. And that was not science. I love science with all my heart. I really do. But I still wanted to stay a dreamer. And so when I was approached by a European university to think about establishing a concept of an international center for aging research, and went and visited for the first time, fell in love with the concept and have been pursuing it now for a couple of years. And I have tried to do it all in high heels. Gentlemen, that is not easy. We all, ladies, try to do that, and that, we're paying the price. And I maintain my shoes in high heels, and I hope I remain in them for the next 20 years as well. So this is what I would like to tell you about. It's really about my latest discovery, my latest passion. I am from Slovenia. I slowly started going back after a while because I was invited to give talks. And when I was approached with the concept of establishing an international center for aging research, the university's president took me to the site and said, well, this is potentially a site where we would establish the center. And it was a 16th century palace. And we have interest on the side of European Union because it was part of cultural heritage in Europe and would potentially come with significant European funding for restoration of the entire complex that contained frescoes and wonderful, wonderful things that could be saved. It really all came together. Still, the university that recruited me had absolutely no history in biomedical research. They're great particle physicists. They knew nothing about biology, but they thought it would be really cool to have this. And so they said, why don't you make it happen? And it's really, this was a whole new challenge. I have never done anything like this before. So this is a mix of science. This is a mix of, if you wish, entrepreneurship, because it's development. It's really tying together people from all over. We have heard today how important it is to have interdisciplinary approach. This is all this is. And most importantly, what struck me really to find, how do I ground this project in this area at all? And this is an area of Slovenia where they have a rich tradition in winemaking, in olive oil making, all the foods that traditionally have been associated with anti-aging compounds, antioxidants. I figured that maybe this is the title that Brussels would really not object to when we tried to sell them the concept as if we knew what we were doing. So it's where sun, soil, and tradition join science for healthy and active aging. And in the context of this palace that I'm going to show you a little more about, I think it really makes a very convincing sense. And maybe next year or year after, I can tell you how this is going. Yeah.
Absolutely. I just wrote it down.
Yeah. And so this is really what grounded the whole thing. I said, okay, well, they might not have biology, but they sure have wine. They have a lot of wine of all denominations. They have cherries, another food that's really high in anti-aging compounds and polyphenols, olives, and the site is absolutely spectacular. So for my recent talks, I'm really trying to sell, to really sell to people, not for money, but in terms of a concept that yes, all the science that we do is really great. And being in regenerative medicine and stem cell biology, we have really, as a group, made a lot of discoveries and a lot of good will come from it. But I think it's equally important, if we're developing these therapies, to put them into people when people are at their best possible state to receive them as treatment. So it goes into this overall well-being. And if it's involved with wine, cherries, and olives, I don't think many of us would complain, even if it's not that effective. But I really do think that overall well-being is probably a better receptacle for any kind of medical intervention than it is when your body is worn out and you're down and you're miserable. And we'll see how it works. But the 50 million euro proposed center is an effort of 8 groups so far in Slovenia. All 8 are very different. We have a strong group in biomaterials and materials in general. We have a group in computer modeling. We have biomedical engineers, biologists, chemical engineers. Just like we've heard repeatedly today, without all those components, this research that we propose that will focus on various interventions for improving the quality of aging, not stopping aging, not extension of human life, but really improving the quality of aging in a sense. If I can be 80 and not ache and not be dependent on a lot of drugs or interventions, I think that would be a very, very good achievement for humankind. We tie that with a strong translational, of course, component. We have local companies that are very interested in helping us identify the main need locally in Central Europe for translation into products. Of course, it comes with a couple of new educational programs that are going to tie the whole concept of anti-aging medicine together. We would like to combine it with the well-being, as I said, and do it all in the 16th century castle if possible. And hopefully this is something that we will be able to show after it's all said and done. So really quickly, why do we age? Well, we age because really for 2 main reasons. One is that oxidative damage to our DNA and RNA to our lives eventually accumulates to a critical mass where cell function is affected. And the second reason is that through repeated cell replication and division, telomeres on the chromosomes get shorter, and after a critical size has been shortened, the cell is not able to replicate anymore, known as replicative senescence. So together between the DNA damage in the shortening telomeres, we unfortunately cannot escape this progression in chronological age. However, we can intervene and minimize the damage that is going to happen one way or another. If cells die for particular reasons, we are obviously tissue engineering and regenerative medicine is developing strategies for replacement of lost cells. A lot of stem cell-based therapies are geared towards replacing cells that were lost, such as Parkinson's, diabetes, and others. I think this one is really critical, and I think we need to spend a lot more time on. It's trying to figure out how to stimulate endogenous tissue to heal itself better, instead of forming non-functional scars or leaving a void, is really waking up this innate ability that has to exist somewhere. It does. We all have adult stem cells. So there is something that can be woken up to help us endogenously regenerate better. Combating cellular events, we're going to do that with wine and olive oil, hopefully, effectively. And removal of aging cells is really something that is not talked about a lot. So removing cells is maybe a targeted goal sometimes in cancer, but this is really not being discussed much. If we remove aging cells or cells that are becoming senescent in our various tissues, by their removal, can we remove the environment that they create that is not friendly for the rest of the tissue? Would that help? Just removing cells that are on the way out and clearing the environment for the rest of them to survive better. And aging changes don't happen when you're really, really, really old. They really start, a variety of them. This is from a review at 45 and so on. The earliest one obviously being evident in the skin, but it becomes progressively worse with all our tissues and with all our systems. So strategy one, I mentioned 4 different strategies. First one is replacing lost cells with new, young, functional cells, preferably autologous if we can, so from our own source. This is not news to anybody. The loss of cells is really a cause of a number of degenerative disorders, as well as traumatic injuries, and therapies for this are being developed as we speak. The second one is stimulation of endogenous regenerative potential. And it's really the fact that we have organisms that can do this on a regular basis, such as salamander. Axolotl is pictured up on top right corner, and probably seen this picture on the left. Regenerative response in axolotl after amputation of a limb. Over a period of about 40 to 50 days, the animal is able to regrow the entire limb to the perfect size again, like nothing happened. And this is not the only example. Starfish, we have all seen those. If they lose an arm, not only does the starfish regenerate a missing arm, but the little missing arm piece will regenerate the other 4. So regeneration is not a new concept. It happens. It just doesn't happen in humans for a number of reasons. One is strong immune response, and the second, or inflammatory immune response. The second is really that our bodies are more concerned in surviving than functioning, so we tend to shrink the wound site, heal it, congeal the blood, form a covering, and then deal with the consequences so we don't bleed out. And those conditions that help us survive are really not friendly to regenerative response. Other animals have chosen different outcomes. But funny enough, this response, complete regeneration, and this likely response after amputation in a human is really mediated by the same cell type in both animals. It's a skin fibroblast. It's a fibroblast that resides in an area of an amputation. It's a most prevalent connective tissue cell. It just so happens that here it can remember what is missing, and it doesn't care what's missing over there. It acquires completely different functions. So my lab is really spending a lot of time trying to figure out, can we wake up this regenerative memory? Memory is clearly there. All our cells are the same. So how do we go fishing for this magic memory that would allow this kind of response in human? Wouldn't it be cool? That'd be kind of nice to do that. not just lizards and starfish, but humans. That would be really very useful. So over the past few years, we have played with our cells in a number of ways to try to modify who they are in culture. And we can make our cells start expressing stem cell specific transcription factors without addition of transgene. I'm not talking about reduced pluripotent cells. We have induced selectively expression on some very primitive neuronal markers in our cells just by manipulating the environment in which we grow cells, which would make all the sense if you think about it. The cell is what it is because of the environment in which it resides. It's not the cell that decides what it's going to be. It's probably its collective environment that's instructing it what it needs to do. So by changing the environment in a very defined way, we have really managed to develop the cells that we have started testing for this hopefully regenerative capability, or more of a plastic capability in a skeletal muscle injury. Those are massive injuries, and we can't treat them with cells alone. So what we do is we seed cells into fiber and microthreads to give them three-dimensional structure. We can organize these microthreads in various configurations to build a volume with enough porosity for the cells to stay alive. And then when we transplant this kind of construct, these are threads, cross-section, into a mouse massive skeletal muscle injury and watch it over time, the final outcome in 10 weeks is really uncontrolled scar. The blue is all collagen, and treated muscle after implantation of cells that we have provided on fiber microthreads. And so this was really encouraging, saying a couple of things, really. It said this could be a consequence of the cells that we have put in. They have made us miraculous muscle back. Or we have really caused the tissue in itself around to respond different to what we have provided, or both. But it's clear that scar deposition in our treated animals is way down compared to untreated controls that are allowed to heal on their own, or are allowed to heal with only the threads present without cells. And to look whether this is really due to whatever we were putting in, we first looked whether after transplantation we were retaining any human DNA. So we are using human cells, putting them into mice. We should be able to track them according to human markers. And we do see the long-term engraftment 30 days out of human cells in this tissue. However, it becomes quickly obvious if you look for the human nuclear antigen that the majority of the tissue that has regrown is not due to human cells. It is in some areas, even the muscle. Dark nuclei is human nuclear antigen, but the majority of the tissue that has filled the wound comes from the mouse, which is really, really exciting. Even though our cells, the cells that we have transplanted as well, stain positive for satellite cells, which are muscle stem cells. So we think they're able to provide both functions. And we do think also that that's dependent on, again, and they're directed by the environment that they are going in. Okay, strategy 2. So we think we have developed, a neat enough approach with regeneration competent cells that, by the way, don't form tumors instead mice that may be useful for tissue-specific regenerative response. So this is how I'm trying to now justify the wine and the olive oil to people who are listening. I know nothing about antioxidants, so this is a whole new area for me, and I rely on my friends to provide me with slides and to explain to me why we can use this to base the entire center in Vipava. And we've heard that too. The aging that results eventually either in disease or cell death, it's really during your lifetime a consequence of many, many, many different factors. And a lot of them are the consequence of the generation of free radicals that do a number, obviously. So if we could develop strategies that could combat all sorts of conditions that are caused by free radicals through either high production in local foods, increasing delivery and bioavailability of these antioxidants. I think we might be doing our bodies a favor and preventing some of these aging diseases as well. You have to like these. These are pretty pictures, even when you make slides. This is good. I like just about everything here. And it's really, this is not new, and I don't think all of it is hocus pocus. A lot of antioxidants have been described as anti-aging magic drugs for a long time. Some of it is very real. For example, stomach cancer, I think it was in the maybe 60s, 70s, almost completely disappeared in the United States, and that coincided, and that's a story, I don't have any factual data that there's a correlation, but coincided with a time where cereals were supplemented with antioxidants. And then they stopped the supplementation after a while, and the cancer is gradually coming back. That might be a coincidence, but those were studies that were published, for sure. So antioxidants for sure cannot be bad, number one. Number two, the problem, however, with them is that they just don't get into cells at the concentrations that could combat one-on-one free radicals. That's the problem, is really getting the concentration in the cell to a high level of enough where they can be relevant. But they do, if we just focus on some of the food where it has been described. You know, I like both these, and I think I'm in a good group because you don't need that much of this to really achieve the quantities and bioavailability of antioxidants that you would get with the juices from various other sources. So it's really, in the university that asked me to put this together, it's really, they have a department where they study polyphenols that are acquired from wine and from olive oil and from other fruits and vegetables. So there's a relevant expertise available on non-flavonoids and flavonoids that are present in all this, and as well for the synthesis, if needed, of any of these compounds. And the majority of the studies right now are being focused on resveratrol, and you probably have heard through the news about these anyway. Again, this is not my field, and I really rely on people who work with polyphenols. And the last strategy is really removal of aging cells. One way is using nanoparticles. We have heard about nanoparticles, and some of the main concern is that they are toxic. Well, maybe sometimes we would want them to be toxic, especially if we want to target cells for removal. So it's not necessarily taking advantage for both things. So you can use nanoparticles to deliver now drugs to targeted cells within the loaded nanoparticle, or you can use it to specifically target cells and deliver a cytotoxic drug. So it's either way. But how do we target cells? Where do we want this to go? We're developing a brand new approach for that that's going to be based on a complex expression of surface molecules and VHH screening in order to be able to target these guys to go to either cancer cells for elimination or to local other cells for delivery of antioxidants. And this is after renovation of this dream that started 2 years ago. The first part of this complex has been renovated. This is one of the main buildings. The palace, this is a river, one of the parts of the river that has 7, I don't know how you call them, individual smaller rivers that contribute to a bigger river eventually, flows through the entire complex, and it looks absolutely fantastic. These are the frescoes that were left and were restored. The ceiling that you see is original 16th century wood beams that have been taken out individually, refinished, and put back in. And this is where laboratories are going to be. This is the space where actual labs are going to be. My lab included. And this is how you get to the lab. I mean, you can't appreciate it on pictures half as much as you could in person. And you are all invited once we get this going, but it's a gorgeous environment that I hope is going to be just as stimulating to the scientists doing science as it is to the visitors. And so with this dream that I'm really happy to be able to work on, I do continue to redefine my success, I think, every day and every year. I think it's beautiful to be a woman scientist, to be able to be a woman scientist in this day and age, and I sure hope I can continue doing that for quite a while. Thank you.
I accept your invitation.
I will just send it to Marianne. She will know where to find everyone. Yeah.
You mentioned healthy aging.
Yeah.
And you sort of said that not longevity. But if you're successful in healthy aging, then you'll achieve longevity. Is there something wrong with longevity?
No, but it's not the main purpose, really. You know, if that's a consequence, that's okay. But I think the goal is more, and the goal is more really removing the effects of aging and really not dealing when you're 70 and 80. I don't think we're ever going to be able. So, for example, our cells, we can double their proliferative span, but it's not immortal. But they look fine. They don't senesce as early, so they will go longer. But at least they look better. So we'll take it. I think that outcome as well.
So
Yes.
one thing that I found particularly heartening about the way that you structured your institute is that so much of the work is actually regenerative, actually repairing damage rather than simply slowing down the pre-existing damage. It is a bit of damage to go in the future. Really only number 3 out of your 4 categories constitutes the slowing down of aging rather than the reversal of the aging damage. And I think that's probably the thing that makes your institute unique among institutes around the world that are working on this sort of problem. Did you do that on purpose?
No. I was just about to say they were in no particular order. The reason I did it in that way, which might be advantageous, is really that I'm least comfortable with it because that's not what I'm proposing to do myself. It's other people. So I started with things that I'm a lot more comfortable with.
Sorry, no, I wouldn't mean the order in which you said the things. I mean the fact that most of what you're doing is regenerative. No.
Oh, okay.
Yeah, I mean, like the Bucking Institute in America, in California, is almost entirely focused on slowing aging down rather than on repairing damage.
Right.
My work is very much focused on repairing damage.
Right.
So I can add a few things to your list. It's a damn good list to be starting with.
Thanks. I think doing everything essentially in parallel may have an advantage, and this place will have the capability to really, dovetail as we go. With all of it. Yes.
What do you see as a challenge to set this kind of research institute in specific location you're thinking of? I mean, it's pure influence of it.
You mean for this particular one? The biggest challenge is, they occur daily. It's really, my next big headache is the complex where this resides is a complex that has 120,000 square feet. They renovated, made a quarter of it, and now we have to compete again within the European Union to continue renovating the entire part of the complex that will house more research labs, the wellness center, the wine institute, the enology labs, and so on. So funding is always problematic. I have found, though, that getting people seriously interested, especially scientists and researchers, and businesses to pitch in the effort for this kind of concept was very easy to accomplish. We have a number of groups in Italy that are eager to design the components, to provide expertise, to financially support some activities that are ongoing. So interest has been easy, and willingness to do it. It's really logistics of, this is not a small effort. This is going to be hopefully, an all-inclusive facility one day, and if money stops for renovations, then what do you do? That's.
How about people?
No shortage. No shortage of people, even from fields that traditionally are not associated, because I think they can all identify. I think today's talk, first in the morning, set the stage perfect. there's no one-man science or one-man solution. We have big and complex problems that require complex teams to be put together with material scientists, chemical engineers, computer, so you name it, right? And so no matter who you talk to, they themselves can find, well, we could belong in here, and this is what we could contribute. I had computer group approach us and say, well, you know what, we're really developing some interesting new algorithms for modeling blood flow. Great. I don't know what I'm going to do with it yet, but it's really, when you define a name like, you know, for healthy aging, you say healthy aging, it quickly becomes clear that anything, almost anything, can be part of this kind of endeavor within the plant medicine.
What exists now that you would see as most similar to this? Clearly not like it, but what do you see that exists now that would be most similar?
I think Aubrey would be a better person to have that, but from my personal experience, not necessarily in aging, I would say Woods Hole. I catch myself thinking, I would like this concept to be Woods Hole for aging research. And those of you who have been at MBL, you know exactly what I'm thinking. You come with flip-flops and shorts, and there's some great science being done all the time by groups that just spend time at the same time within the same space, thinking about similar problems. But I don't know what would be. Yeah.
I mean, I would have probably said the Bucket Institute, but I would be working from the other end. You know, they're an amazing institute, but their environment is not by that day scale. Of course, their building is just as spectacular in its own very different way.
Right.
But yeah, I mean, I think from the point of view of the atmosphere and the culture that you're trying to create, I think Woods Hole is a great example.
Yeah. Thanks. Yeah.
And what about the role of genetics? Is that being studied at your institute, given that we know from genetic research, some of which is going on in Einstein actually, with individual families who have very long longevity and finding genetic markers, and how that's applied to this research?
Yeah, I haven't even talked about the scientific content. Yes, the region where this is, we have already identified a number of families that are 4 or 5 generation families already. It's not quite typical for the region, but it's part of Mediterranean where that has been described before. And the whole neurobiology group that is dealing with neurodegenerative diseases is proposing exactly what you're talking about. It's really genetic screening based